So I'm going to change completely gears from disease state to something that is not a disease. It's something that everybody, the whole world is expected to go through. And actually, maybe it's the lucky ones who do get through and not some young person who may would not have the chance to do that. But it's about aging, and it's something that people have considered as not a disease, but something that is not wanted. My background is in biomedical engineering. I'm not a dermatologist, but I have worked a lot with dermatologists, and I've done a lot of works on skin, mostly actually with babies. So most on the early life. What I'm going to present is more about later stage in life. And I would say some new perspectives and new paradigm shifts. Of course, a lot of work has been done on aging. We talked about cosmetic dermatology yesterday. You know, this is correcting some aspects of aging. But I'm going to discuss a little bit different perspectives. I work for SGS, which is a very big company, but not very known to the whole world. I used to work for Johnson & Johnson before, and everybody knows Johnson & Johnson. SGS is as big as Johnson & Johnson, but they are B2B. Basically, it's a service organism for pharmaceutical, actually for the whole industry, but part of it is on healthcare and skincare specifically. We do clinical trials for pharmaceutical and cosmetic industry. And while we do that, we develop non-invasive methods to be able to measure things quantitative and objectively on skin. So go quickly through that. I know we're a little bit late in time, so I will try to go quickly. This slide, most of us know aging relates to, depending on the ethnic background, and yesterday we had a nice talk on diversity, can relate mostly to surface features like, surface structure like wrinkles, but it could also relate to pigmentation and differences or inhomogeneities in pigmentation. In certain people of certain background may be more on dryness and radiance, or more on age spots rather than wrinkles. But a lot of work has been done on understanding that. We talk about extrinsic and intrinsic aging, depending on what are the features, the exposures that can lead to the aging phenotype. So with intrinsic aging, this is the green bubbles there. We have genetics, we have hormonal changes, we have natural or time-related cell senescence. We also have the extrinsic, which relates more to how we live, what is the lifestyle, what do we eat, whether we smoke or not, whether we are exposed to the sun or the pollution. And these kind of exposures accelerate aging features. So it has been a few years now that this idea of healthy aging has emerged. And this is one of the concepts that I would like to speak to you today, which is not so much to go against the science of aging, but more about how a person who is aging feels. How do we make sure that they feel healthy and content and happy? So there was an article that was published not so long ago with this short concept, which is, it's not the number of years that matters, but it's the quality of life. How do we live? As a parenthesis, I have done some work on dermatological side effects of oncology patients, oncology therapies, and actually patients there, they care more about the way that they live their life rather than if they can have 1 or 2 more years based on, having drugs and with the quality of life going down. So quality of life is very, very important to patients and to all people. So what can we do to improve this quality of life and reduce the effects of aging? This paper here was focusing on reducing the oxidative stress. This is the imbalance in oxidative stress can lead to accelerated signs of aging, but also to several diseases like cardiovascular diseases, cancer, diabetes, and so on. So a particular focus initially was pointed to oxidative stress, and for that, to target that, could be, to address that, we could look at biology and mechanistics, which means more drug therapies like mTOR inhibitors and hormone. I'm sorry for the spelling mistake. I thought that I corrected. Like hormone therapy and telomere dysfunctions, treatments for telomeres. So this is quite pharma approach. I would say it's not really. You're not talking about something that all people can do daily, which would be more like a cosmetic effect. So besides the oxidative stress, or actually what is the regions that can trigger oxidative stress, there is this psychological aspect. And we talk about psychology, that the psychological stress that can trigger immune dysfunction can trigger measured reactive oxygen species production, DNA damage, and so on. So how can we intervene there is through lifestyle, and of course avoidance of environmental factors that can accelerate the production of oxygen species, reactive oxygen species. So I talked about pharma, but how can we do this with cosmetics? And cosmetic treatments can be used not only to target a specific feature, but also to make a person feel a little bit better. I put these pictures here because when we talk about cosmetics, we think about beauty, and beauty is not necessarily just linked to how young a person is. So I have a picture here of an older lady and a younger lady, but the way that they feel and the way they express their feelings, I think if you say who is more beautiful, you would say the one that's smiling. It's a no-brainer. So it's the way that we feel also that plays a role in beauty, and of course in what cosmetics can do. So how can we measure actually a psychological effect of a cosmetic? This is an active area of research right now. We can measure electrochemical signals from the skin, like with electrodes on the finger. But more recently, we've seen the rise of small EEG devices, electroencephalography devices, that can be used in a clinical setting for a clinical trial to see what part of the brain is activated when a person does something, including smelling a cosmetic or putting a cosmetic on their face. So there are 2-3 companies now around the world that make these devices that have only a few electrodes or a little hat that you can put on while you're in a clinical trial, and you can measure activation of parts of the brain that can link to valence, means how intensely the person is feeling, and the arousal rates, whether it's something that is positive and negative. So this is very, very basic. Of course, we're not going into looking in high resolution, but more trying to correlate the feeling that is assessed typically with a questionnaire with something more objective that is looking at which parts of the brain are activated. And we have some examples of that for different interventions using scented cosmetic products. Besides the EEG, other tests that we can do is to measure cortisol levels in saliva. So when we do cosmetic testing, always we have to be aware that it has to be non-invasive and as mild as possible so that it's ethically approved. So cortisol levels is a swab of saliva and then measuring with ELISA or amylase levels. So there are these techniques that are being developed right now that can have a more objective measurement of on the feeling of the person when using cosmetics. The second concept that I want to talk to you today is this pre-aging concept. This actually doesn't concern so much the older categories, but more the middle categories where they start seeing some early signs of aging on the face. And sometimes, to me, it was surprising when I first saw it, but apparently it's a very, very big trend, especially in Asia and in East Asia. That's where it started growing, and now it has spread to other parts of the world. So the concept is how can we prevent signs of aging from happening? So to prevent, first we have to characterize what is the signs that we want to measure. So decrease elasticity, fine lines, wrinkles, pigmentation that I mentioned before, and skin dullness. And then what are the contributing factors? I mentioned genetics and psychological stress, the environment and lifestyle. So we have to understand how these factors are affecting the biology and how biology would be eventually expressing signs of aging. So one of the main characteristic for that happens early, and this is where we talk about pre-aging, is the slowing down of the cell turnover rates. And I'm talking about the epidermis now and the basal layer. And this can happen because of over time accumulation of mutations, misfolded proteins, and overuse of mitochondria. So the general idea is that reactive oxygen species can trigger inflammation, and then eventually inflammation can lead to slowing down of the cells, these senescent cells that eventually would not be functional, or at least partially decrease their main function. So such mechanisms can provide targets for cosmetics, for skincare actives. Now, this is a schematic basically summarizing what I've said before in the previous slide, is that reducing capacity of DNA to repair itself can lead to increased production of metalloproteinases, which would decrease collagen production and have other collagen changes like accumulation of advanced glycation end products and thickening of fibers, but loss of new collagen deposition, which can lead to color changes like sallowness and anisotropy of fibers. DNA repair, the reduction in DNA repair capacity can increase cellular stress, which increases cell cycle arrest and pro-inflammatory signaling. Eventually, this is, I think, the most important decrease in cell proliferation, and the increased production of cytokines. So cytokines, typically we think about inflammation, but there are a lot of cytokines that are normally there on skin, acting as innate immunity as our first line of defense. But when we have a disturbance of the balance, then we can have an increased production of these cytokines. So I was talking about non-invasive methods. One of the methods for measuring cell turnover rate is to look at tryptophan fluorescence. And there are spectrometers with fiber optic probes that you can bring in contact with the surface of the skin. You can measure fluorescence. Now, this fluorescence, there are different bands. There is one band that is characteristic to tryptophan. And one of my early mentors had demonstrated in the 80s that these bands is relating to turnover rate. Now, we did a study about 20 years ago in Asia Pacific in different areas and in different times of the year. This is an aging study, so you get a lot of healthy people at different ages, and then you measure fluorescence, and then you see if there are correlations. The amazing thing for me was that, okay, yes, there is a correlation with tryptophan fluorescence, so there is a decrease in the cell turnover rate. But the amazing thing is that the slope is pretty much the same, independent of the area that we were studying, where we collected the data, and independent of the season. So when we talk about the evidence of pre-aging in a young population is that these people down here that have low turnover rates already in their 80s to 30s. So there are young people here that have healthy turnover rates, and then this turnover rate is reducing with time. Now, the question for cosmetic is how can we increase the turnover rate so that they have renewals in the, cell renewal, and don't have accumulation of the effects that I showed before. Another aspect, again, with non-invasive method is we can look at skin close-up, it's like thermoscopy, and we can measure the mechanical forces if they have a particular direction or not, and then if they don't, then they have a low anisotropy, and if they have they do have particular direction, then they have higher anisotropy rates. And this anisotropy is related to aging. So in this case, when we talk about pre-aging, it's how can we maintain a low anisotropy ratio? And this is basically by keeping collagen production to continue at the young rates. And as you can see, I'm talking more about how do you measure things rather than actives or methods of intervention. And finally, the third insight from non-invasive methods is imaging, and then evaluation of imaging, and how different parts of the image can affect the perception of aging in the general public. And this is a publication that we had in 2008 showing that there are a lot of different features, but we can focus on 5-6 features that are the ones that affect more the perception of aging. And of course, this is origin dependent, and also there is an interesting age bias. So basically, people who are grading their own group are more sensitive than grading a different age group. So there are different methods that I showed, besides those methods, there is also three-dimensional analysis of wrinkles. So there are a lot of methods that can measure quantitatively and objectively different aspects of aging and even early signs, these preclinical signs of aging. For cosmetic companies, they are interested in what claims you can put on a new ingredient. So they can talk about brightening or reduction of dark spots and so on using these quantitative objective methods. So in summary, healthy aging, there is new emphasis on well-being. This is the aspect of healthy aging. We need to have ways of measuring the effects of interventions on that. Same thing for pre-aging. We need to have ways of measuring early signs. So the methods have to be, besides non-invasive, they have to be very, very sensitive and to measure something that hasn't appeared yet. So it's a little bit a play of prediction. I haven't talked about the role of AI, which is something quite promising also to be able to, based on a large amount of data that can be collected, how you can predict what is the first sign on a specific, on a person with a specific background. This is very much active area of research. And with that, would like to conclude my presentation. Thank you.